Adsorption of hexavalent chromium using tamarind fruit shell. Abstract, to investigate the biosorption of chromium, 6, ion using crude tamarind fruit shell, CTFS, and HCl treated tamarind fruit shells, HTS, from aqueous solution at room temperature to compare the extent of biosorption between untreated and treated forms of the tamarind shells to evaluate the influence of different experimental parameters such as pH, effect of initial metal ion concentration and effect of dosage of adsorbent on biosorption. Introduction. Heavy metals are naturally occurring elements that have a high atomic weight and a density at least 5 times greater than that of water. Their multiple industrial, domestic, agricultural, medical, and technological applications have led to their wide distribution in the environment, raising concerns over their potential effects on human health and the environment. Their toxicity depends on several factors including the dose, route of exposure, and chemical species, as well as the age, gender, genetics, and nutritional status of exposed individuals. Because of their high degree of toxicity, arsenic, cadmium, chromium, lead, and mercury rank among the priority metals that are of public health significance. These metallic elements are considered systemic toxicants that are known to induce multiple organ damage, even at lower levels of exposure. They are also classified as human carcinogens. Due to their persistence in nature, it is inevitable to become essential to remove them from wastewaters. Conventional techniques have limitations and often are neither effective nor economical, especially for the removal of heavy metals at low concentrations. New separation methods are effective and environmentally acceptable at affordable cost. An alternative method for the removal of heavy metals is considered based on the biological methods of metal removal and recovery. Microorganisms play a significant role in bioremediation of heavy metal contaminated soil and wastewater. The ability of microorganisms to remove heavy metals from aqueous solutions has been known for some decades. The removal of the metals occur actively only with living cells and or passively at the surface of dead cells. This is known as biosorption. 2. Biosorption is a fast and reversible reaction of heavy metals with microorganisms or biomass. In this study, heavy metal-resistant fungi and bacteria were isolated from the soil samples of an electroplating industry, and the bioaccumulations of Cr6 by these isolates were characterized to evaluate their applicability for heavy metal removal from industrial wastewaters. The optimum pH and temperature conditions for both the growth and heavy metal removal were determined for each isolate. The optimal pH for fungal isolates was lower, 5.2, than that for bacterial isolates. The observed effects of pH was attributable mainly to organism-specific physiology because in all the tested cases the cellular growth positively correlated with heavy metal removal. The byproducts obtained from biomaterial production are cheap source of biosorbents. Therefore biosorption is potential cost-effective way of removing heavy metals from industrial wastewater. The process is effective even if the concentration is as low as 200 g/ml. Several approaches have been studied and developed for the effective removal of heavy metals using biosorbent like microbial biomass and other agricultural byproducts: soybean hulls, walnuts hulls, cottonseed hulls and corn cobs. It has also been observed that these biosorbents need further modifications to increase the active binding sites and also made them readily available for sorption. Huang and Huang stated that pretreatment of biomass removes the surface impurities on the biosorbents and expose the available binding sites for metal sorption. 5. Toxic effects of chromium. Chromium was discovered in 1797 by Vauquelin. Numerous industrial application raised chromium to very important economic element. At the same time with the development of its uses, the adverse effect of chromium compounds in human health were being defined. Chromium as pure metal has no adverse effect. Little toxic effect is attributed to trivalent chromium when present in large quantities. Both acute and chronic toxicity of chromium are mainly caused by hexavalent compounds. Several in vitro studies indicated that high concentrations of chromium, 3, in the cell can lead to DNA damage. Hexavalent chromium is also one of the substances whose use is restricted by the European Restriction of Hazardous Substance Directive. The carcinogenity of chromate dust has been known for a long time, and 1890 the first publication described the elevated cancer risk of workers in a chromate dye company. 3 mechanisms have been proposed to describe the genotoxicity of chromium, 6, to chromium, 3. The first mechanism includes highly reactive hydroxyl radicals and other reactive radicals which are byproducts of the direct binding chromium, 6, produced by reduction in the cell to the DNA. The last mechanism attributes the genotoxicity to the binding to the DNA of the end product of the chromium, 3, reduction. Overexposure to chromium can occur in welders and other workers in the metallurgical industry, persons taking chromium-containing dietary supplements, patients who have received metallic surgical implants, and individuals who ingest chromium salts. Chromium concentrations in whole blood plasma, serum, or urine may be measured to assist in the forensic investigation in the case of fatal overdosage. Since plants lack a specific transport system for CR, it is taken up by carriers of essential ions such as sulfate or iron. Toxic effects of CR on plant growth and development include alterations in the germination process as well as in the growth of roots, stems and leaves, which may affect total dry matter production and yield. CR also causes deleterious effects on plant physiological processes such as photosynthesis, water relations, and mineral nutrition. Metabolic alterations by CR exposure have also been described in plants either by a direct effect on enzymes or other metabolites or by its ability to generate reactive oxygen species which may cause oxidative for safety in exposed workers, to confirm the diagnosis in potential poisoning victims, or to stress. The potential of plants with the capacity to accumulate or to stabilize CR compounds for bioremediation of CR contamination has gained interest in recent years. 3. Adsorption. Adsorption refers to the existence of a higher concentration of a molecular species at the surface of solid or liquid material than is present in the bulk of the material. The material attracts and retains other species on its surface is called the adsorbent, and the species that gets retained on the surface of material is called adsorbate. I.e., the adsorbing material is called adsorbent and the material that gets adsorbed is called adsorbate. The process in which an adsorbed material leaves the surface is called desorption. Adsorption is different from absorption. In absorption, the molecules of a substance are uniformly distributed in the bulk of other, whereas in adsorption molecule of one substances are present in higher concentration at the surface rather than bulk of the other substances. Adsorption isotherms. The process of adsorption is usually studied through graphs known as adsorption isotherm. It is the graph between the amounts of adsorbate, X, adsorbed on the surface of adsorbent, M, and pressure at constant temperature. Different adsorption isotherms have been Freundlich, Langmuir, and Bet theory. A. Basic adsorption isotherm. In the process of adsorption, adsorbate gets adsorbed on adsorbent. According to Le Chatelier principle, the direction of equilibrium would shift in that direction where the stress can be relieved. In case of application of excess of pressure to the equilibrium system, the equilibrium will shift in the direction where the number of molecules decreases. Since number of molecules decreases in forward direction, with the increases in pressure, forward direction of equilibrium will be favored. Fig. 3. Basic adsorption isotherm. From the graph, we can predict that after saturation pressure Ps, adsorption does not occur anymore. This can be explained by the fact that there are limited numbers of vacancies on the surface of the adsorbent. At high pressure a stage is reached when all the sites are occupied and further increase in pressure does not cause any difference in adsorption process. At high pressure, adsorption is independent of pressure. B. Freundlich adsorption isotherm. In 1909, Freundlich gave an empirical expression representing the isothermal variation of adsorption of a quantity of gas adsorbed by unit mass of solid adsorbent with pressure. This equation is known as Freundlich adsorption isotherm or Freundlich adsorption equation or simply Freundlich isotherm. where X is the mass of the gas adsorbed on mass m of the adsorbent at pressure P and K, n are constants whose values depend upon adsorbent and gas at particular temperature. Though Freundlich isotherm correctly established the relationship of adsorption with pressure at lower values, it failed to predict value of adsorption at higher pressure. See Langmuir adsorption isotherm. In 1916 Langmuir proposed another adsorption isotherm known as Langmuir adsorption isotherm. This isotherm was based on different assumptions, one of which is that dynamic equilibrium exists between adsorbed gaseous molecules and the free gaseous molecules, where A, G, is unadsorbed gaseous molecule, B, S, is unoccupied metal surface and AB is adsorbed gaseous molecule. Based on his theory, he derived Langmuir equation which depicted a relationship between the number of active sites of the surface undergoing adsorption and pressure, where the number of sites of the surface which are covered with gaseous molecule, P, represents pressure and K is the equilibrium constant for distribution of adsorbate between the surface and the gas phase. The basic limitation of Langmuir adsorption equation is that it is valid at low pressure only. At lower pressure, Kp is so small that factor 1 plus Kp in denominator can almost be ignored. So Langmuir equation reduces to equals Kp. At high pressure Kp is so large that factor 1 plus Kp in denominator is nearly equal to Kp. So Langmuir equation reduces to tamarind fruit shell. Tamarind indica, consist of fruit, black seeds, and husk. Tamarind husk is outer shell of tamarind fruit obtained after removing the fruit. The process for obtaining tamarind husk is during the separation process to remove the fruit and the black seed. Tamarind seed husk is brown outer shell of black seed obtained after decortications of roasted seeds. It has wide uses in fuel and cattle feed industry, and also tannins extracted from tamarind husk are used in leather industry, and tamarind husk is also used for fish poison. Tamarind indica fruit shells, TIFSs, are naturally calcium-rich compounds. They were impregnated with ammonium carbonate and then carbonized, leading to ammonium carbonate activated ACATIFS carbon. The resulting materials and carbon arising from virgin fruit shells VTIFS were characterized and assayed as adsorbent for the removal of fluoride anions from groundwater. Tamarind husk and seed husk uses are not limited to no applications and usages, but it has many unknown usages and applications, too. Fig.6. Tamarind fruit. 2. Materials used. Tamarind fruit shell, waste product of tamarind pulp, were collected from the natural tamarind trees, HCl, NaOH, K2Cr2O7, Morse salt, 2NH2SO4 etc. Methods used. A. Preparation of crude tamarind fruit shell, CTFS, biosorbent. Tamarind fruit shells, a waste product of tamarind pulp, were collected from the natural tamarind trees. The tamarind fruit shells are cleaned with distilled water, and are powdered and sieved to get uniform size. They are again washed thoroughly with distilled water and dried in the oven for 2 hours at 60 degrees C. It is used as a biosorbent called crude tamarind fruit shell, CTFS. Fig.7. CTFS sample. B. Pretreatment of the biosorbent. The pretreatment of tamarind shells is carried out to increase the metal uptake efficiency. 10 g of the tamarind shells are treated with 100 ml 1NHCl for 24 hours separately, and kept on the water bath, 70 degrees C, for half an hour. It is cooled and is neutralized with 50 ml of 1NaOH. The filtrates are separated and are dried in the oven for 4 hours at 60 degrees C. Thus the pretreated tamarind fruit shells HTS is used as a second and third biosorbent. C. Effect of pH. The effect of pH on chromium biosorption on CTFS and HTS are studied at room temperature by varying pH of chromium solution. The pH of the solutions is adjusted with 0.05 NaHCl and 0.05 NaOH by making use of digital pH meter. D. Preparation of potassium dichromate solution. 1N potassium dichromate was prepared by weighing accurately 12.25 g of potassium dichromate in a 250 ml standard flask, dissolved in distilled water and made up to the mark, shaken well for uniform concentration. From the stock solution, different concentrations like 0.5 N, 0.25 N and 0.125 N was prepared by pipetting 50 ml, 25 ml, and 12.25 ml in 100 ml standard flask and made up to the mark, shaken well for uniform concentration. Fig.8. Potassium dichromate solution. Batch studies. pH of the potassium dichromate solution is adjusted with 0.05 NaHCl and 0.05 NaOH. For the first set of solution, pH is adjusted to 3 and for the second set of solution, pH were adjusted to 5. Batch adsorption experiments are carried out by shaking 1 g of biosorbent mixed with 50 ml potassium dichromate solution of known concentration in each flask solution. Then flasks were shaken thoroughly for about an hour and then kept in a tray containing water to reach equilibrium. After 12 hours the mixture was filtered using Whitman filter paper No. 1. The experiment is repeated with both CTFS and HTS. Fig.9. Batch studies. Preparation of 0.1 N more salt solution. About 9.8 g more salt was accurately weighed into 250 ml standard flask and made up to the mark, shaken well for uniform concentration. Titration of the test sample. 10 ml of more salt and 20 ml of 2N sulfuric acid was pipetted out into a conical flask. 10 drops of N-phenyl anthranoic acid was added as an indicator and titrated against potassium dichromate from burette. At the end point the green color changed into wine red color. Experiment is repeated for all known concentration and pH of potassium dichromate solution. Result and discussions. Percentage of removal with initial concentration. A. Percentage of removal of chromium with initial concentration using CTFS at pH equals 3. Effect of CTFS and HTS. The removal of Cr, 6, is maximum for pretreated biosorbent, HTS, i.e. the adsorptive capacities of the pretreated form are greater than crude form. The treatment of biomass with HCl implies the protonation which displace the lighter metal ions from binding sites, I.e., carboxylic, sulfonic etc. Acid treatment results not only in cleanup of the surface impurities, stabilization of the surface compounds, and increasing the surface area by opening the available sites for metal adsorption. Effect of initial concentration. Removal of chromium decreases with increase in the initial concentration. We can see that maximum removal of chromium takes place at a lower concentration of 0.125 N at all pH. Effect of pH. Solution pH can significantly influence the removal of heavy metals. Here we use two different pH conditions and study the rate of adsorption. It is found that adsorption is maximum at low pH. The decrease in adsorption with increase in pH may be due to the decrease in electrostatic force of attraction between the sorbent and sorbate ions. At lower pH, due to high electrostatic force of attraction, the percentage of Cr, 6, removal is high. At very low pH, the surface of the sorbent is surrounded by hydronium ion which enhances the adsorption of hexavalent chromium. Generation FOF data for Freundlich adsorption isotherm. A. CTFS pH equals 3. The graph log CEVS log x/m is a straight line. From that we conclude that the adsorption of chromium using tamarind fruit shell follows the Freundlich adsorption isotherm. The data is also fit well to Langmuir adsorption isotherm. Conclusions. Biosorbents CTFS and HTS have maximum adsorption capacity compared to many adsorbents. The tamarind fruit shell, both its natural and acid-treated forms, are excellent biosorbents for the removal of chromium ions. The percent of removal of chromium is reduced with increase in initial metal ion concentration. The maximum percent of removal chromium occurred at 0.125 N. The maximum uptake of chromium, 6, ions occurs at pH equals 3. The data fits well to both Langmuir and Freundlich isotherms.